Hello! Hope you have been enjoying your break so far. And if you celebrate Christmas, had a good Christmas. So the first video that we dealt with eukaryotic regulation of genes that occurs before transcription occurs. So at this point, we're at post-transcriptional regulation, and at this point, the mRNA is a mature mRNA, meaning that the mRNA has been spliced, and it has a GTP cap on the 5' end and a poly A tail on the 3' end. And if you remember, just to go through this again, once the mRNA is made, it can be spliced different ways. So depending upon sort of the conditions in the cell can choose to make different things exons and different things introns and make different mRNAs, which then as a result in a different protein. Just to sort of go back to bacteria can't do any post-transcriptional modifications because as soon as the mRNA is transcribed, it's translated. So all this post-transcriptional regulation never is able to occur in bacteria or prokaryotes. So again, here, the point is that once you have a mRNA, it can be spliced different ways, making different parts of the mRNA segment introns and different parts of the mRNA segment exons. And if you have two different mRNAs, that can code for two different proteins, and depending upon the conditions, you may want one protein or the other protein. So once the mRNA is spliced, how long the mRNA lasts in the cytoplasm can determine how much protein is made. And mRNAs in the cytoplasm can last from hours to weeks. And what determines how long they last is the length of the poly A tail. So there are enzymes that break down mRNA that are present in the cytoplasm of all cells, and they break down mRNA from the 3' end. So the longer the poly A tail, the longer the mRNA will last, and the more protein that it will be able to make. Okay, now, the next thing is one of the more complicated things of this lesson. And this was just discovered recently. Oh, sorry, very, 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 very recently. Like I didn't know about this possibility when I was studying biology. So the cell can make special RNAs, which are called small interfering RNAs. Okay, and they're complementary, okay, to the mRNA you might want to destroy. Okay, and if it's complementary to the mRNA, it will bind to it. And the cell recognizes double-stranded mRNA, and it signals whenever it sees double-stranded mRNA, okay, it signals for that mRNA to be destroyed. Okay. Now this has been used in biology to turn off genes. If you want to see what happens in a cell without a particular protein, you can make a small interfering RNA that's complementary to the mRNA, and it will bind to it, as it's doing here. And cells don't like double-stranded RNA, because actually viruses sometimes have double-stranded mRNA, and we don't want viral infection. So whenever a cell sees double-stranded mRNA, okay, it signals, okay, for the cell to cut it up so that it doesn't get to the ribosome. So we have biologists, this happens naturally to control gene expression in the cell, and scientists have actually begun using it to turn off genes in cells and to see what the result is when that protein isn't there. So, unfortunately, I have not been able, I've tried on my school computer, my home computer, I have not been able to get the pen to work. So when I get to school, I'm going to have to ask Corey to help me, because I have not been able to set up or get the driver to install correctly. Sadly, you have to deal with my bad handwriting still. So let's say this is a strand of mRNA. Okay, what they would do to turn off a gene, or what the cell would do, is make a RNA that's complementary to it. So what do we mean by complementary? So if this is like A and this is U, I don't know, and this is G. They would make a small interfering RNA. They're usually about 18 bases long. Okay, that was complementary to this strand. Okay, and what that signals. Okay, so again, this would be the small interfering RNA that you would inject into the cell, or the cell would make, and it would be complementary to the mRNA of the protein that you want to either not be made or you want to turn the gene off. Okay, and then what happens, and this is a little bit of an oversimplification because again, the AP exam doesn't care about our details, is that an enzyme known as dicer, D-I-C-E-R, and when we get to school, I'm going to show you a video that shows the process more completely, comes and says, oh, double-stranded mRNA, it's my job to cut this thing up, okay? And it cuts it up into tiny, tiny little pieces, and then never reaches the ribosome, and as a result, because it never reaches the ribosome, the protein is not made, and that makes the gene actually be turned off. Again, I'm sorry about the pen not working. I don't know. I've tried to install the drivers. I tried it on my dad's computer. My school computer won't let the drivers be installed, so I have to talk to Corey. But we'll see. But anyway, the point is, once the double-stranded mRNA is seen, the enzyme dicer senses this double-stranded mRNA and comes and cuts it up. And then it doesn't make it to the ribosome, and therefore the protein is never made, and the gene is essentially turned off. One of the other ways that you can block or turn off a gene is by blocking translation. So let's say the mRNA makes it to the ribosome. There are regulatory proteins that can attach to the 5' end of the mRNA and prevent the ribosome from being able to hold on to the mRNA. So at the stages, this protein, oh no, all of a sudden we don't want this protein to be made. So this protein basically captures the mRNA, says, up, nope. You're not binding to the ribosome, and then as a result, since the ribosome can't bind, the tRNAs can't bring the amino acids, and the protein won't be made, and then the gene is obviously off, even though the mRNA is made. And sort of the last and final way that we can regulate gene expression is, let's say you've spliced your mRNA, it successfully made it. There's no siRNAs present in the cell to make it get cut up by dicer. There's no proteins that prevent it from associating to the ribosome, and the ribosome translates the protein. And you've wasted all that energy, and now you don't want that protein anymore. The last way to regulate gene expression is if you don't want that protein anymore, so here's that protein that's been made, and you don't want it anymore, the conditions in the cell have changed. Let's say you don't want this cyclin has been made, it's told the cell to divide, and you don't want the cell to divide anymore. The protein can be ubiquitinated, okay, and ubiquitin is a small, I think it's about 70 amino acids long protein. You can ubiquitinate the protein, and that signals for the protein to be destroyed by the organelle which we learned earlier in the year called the proteasome. And now, even though you've made the protein and you don't need the protein anymore, the proteasome will destroy the protein, and then the protein will only be made again when it gets the signal from the transcription factor to be made again. But this is sort of the last stage when you sort of already made the protein and now you don't want it anymore. You can turn off the gene by destroying the protein through ubiquitination. 76 actually amino acids. Didn't know exactly, huh? So again, ubiquitin is a death tag. When you no longer want the proteins that you've made, you can destroy them by ubiquitinating them, and then that signals for the proteasome to come and digest the proteins into amino acids. And then here, this is sort of not important, but this shows the sort of what the proteasome is. It breaks down proteins by about 7 to 9 amino acid fragments. And then those amino acids can then be broken down into single amino acids in the cytoplasm, and then they can actually be reused to make other proteins using the regular translation process, tRNA, bringing them to the ribosome to make any protein that needs to be made. So here, when we get back from vacation, I'm not going to do it now, but I'm going to reference this slide. Whoa. Went too far. I'm going to reference this slide when we get back from break, but this is essentially shows all the ways that you can control genes in eukaryotes. And if you remember, in prokaryotes, we really only had one way, and that was the repressor protein, which will block RNA polymerase from transcribing the DNA. But eukaryotes have many more ways to control gene expression and gene regulation. When we get back from break, we're going to talk about how this process controls development of organisms from a one-cell egg to a full organism is what we're going to talk about when we get back. But on Friday, that's our first class, we're going to review sort of this process, all the ways that gene regulation can be controlled in eukaryotes by going through this slide and adding more notes to it. I'm actually going to print it out in larger size, and we're going to add some notes to it. But enjoy the rest of your vacation. Your slides are due on the 28th, and I will see you guys in about a week.